Okay, I hope everyone is logged in to Moodle. Obviously you are if you're listening to this. As the sub said, you are going to be completing a translation lab assignment, and the goal of this assignment is to understand mutations. So what you'll do once you're logged into Moodle, you should see this screen. You need to click on the transcription and translation virtual lab assignment. As before, you need to right click up here on this green link, open in a new tab, and you should see something like this. Now, it's really important that you guys read the instructions over here, but basically what you will do is you're going to click on the mutate button, and when you click on the mutate button, you're either going to get an original sequence or a mutated sequence of mRNA. So in this case, we got a mutated sequence. Now I'm going to hit it again just to see. I want to start with an original. So here's the original sequence of mRNA right here. AUG, AAA, so on and so forth. And then here are the amino acids that make up the protein based on those letters. So what you have to do is you have to recreate the original sequence. However, you have to do whatever it tells you down here in the mutation rules window. So for this one it says the fifth C becomes a U. So what we do is we just make the same, not matching up complementary letters, we're matching the exact same letters. So we just keep doing whatever it says, just like this, and we have to watch for the fifth C. So I keep doing this, oops, there we go. There's my second C. Just like this, we keep going. Oop, grabbed the wrong letter. There we go. C. So 1, 2, 3, 4. So it's the C right down here that's going to change. Everything else. So basically, you keep going like that, and then for this particular rule, you're going to change this C to a U. All right, and like we talked about yesterday in class when we did the sentence with the big fat cat ate the wet rat, it's going to change the result. All right, so once you put all your RNA nucleotides down here, you're going to have a new protein. Now down here is a chart that you can use. If you don't like this one, you can always just go to Google and bring up the wheel that we use in class like this. Alright, so whatever you want is fine. I guess you could pause the video right here and use it for later if you want to. That would be fine too. However you want to do it. Or you can use this table. You just go first letter, second letter, third letter. So you'd go like U, C, A would be right here, serine. So you can use this one too. It's not bad. Okay, so that's how you basically do this assignment. If you were to get a different one, you get this. All right, this is the mutated sequence, and in this one, the second C was deleted to get this, so that changed everything. So you have to kind of work backwards on these, and if you have a really hard time with one and you just can't figure it out, don't spend a ridiculous amount of time trying to figure it out. Just maybe try another one, all right? But I would like you to do one of each. I would like you to do one where you start with the original and create the mutated, and then I want you to try one where you have the mutated and you work backwards, all right? So if you can't do one, then just try another one. And then once you are all done, you will check it to make sure you did it right. And it tells you down here something that's wrong. And then at the end, of course, you have 6 journal questions that you have to answer and submit. Now remember to submit your answers individually. Even though you guys can work together to find the answers, you need to submit your own. And the best way to do that would be to have one person put them all into their file, into their Moodle submission page, and then just copy those and send them through Moodle to your partner. Copy, paste, and send it in a message. All right? So I hope everything works out well. I will try to keep in touch through Moodle or through Google, and I will talk to you guys on Friday. Thanks, and please remember to be good for the sub.